Hello and welcome to this Introduction to Cell Biology video. I'm Dr. Aled Roberts, and in this video we're going to look at some of the core groups of bacteria that form the human microbiota, where they are found and what role they play in health and disease. Now, when we talk about microbiota, we need to remember that this refers to the community of microorganisms that inhabit a specific environment. It includes all the different bacterial, fungal, archaeal, and protozoal species, as well as non-living entities such as viruses and bacteriophage. And when we are talking about the human microbiota, we are referring to all the microorganisms that live on and within us. So what types of living microorganisms actually dominate the human microbiota? Well, excluding non-living entities like viruses and bacteriophage, bacteria are by far the most abundant, followed by smaller contributions from fungi and archaea. Now, amongst the bacteria, there are four key taxonomic groups that appear consistently as endogenous microorganisms. And these are the firmicutes, bacteroidetes, actinobacteria, and proteobacteria, along with some other phyla. Now we'll go through each of these key phyla in a bit more detail in a moment. However, it's important to note that different phyla, so different groups of bacteria, dominate different body sites. For instance, in the mouth and esophagus, the firmicutes are the most common, whereas the skin is mostly colonized by actinobacteria, with some firmicutes mixed in for good measure. The gastrointestinal tract is roughly a 50-50 split between bacteroidetes and firmicutes. And finally, the male urogenital tract is dominated by proteobacteria, a stark difference to the firmicutes which dominate the female urogenital tract. Now, when we look at higher taxonomic levels, like the phyla we can see here, the microbiota are quite similar between people in terms of composition and abundance. However, at lower levels, so when we start getting down to the species level, there's a lot more variation between two individuals. Okay, so let's look at some example microorganisms that make up each of these phyla, starting with the firmicutes. These are mostly gram-positive bacteria, and they tend to be either rod or cockeye shaped with a diverse range of metabolic activities, allowing them to adapt to a very different environments within the body, resulting in aerobic firmicutes which thrive in the presence of oxygen and allow them to grow on the skin, whilst others are anaerobic, preferring environments that lack oxygen such as the GI tract. Now, within this phylum there are two particularly notable orders of bacteria, the bacilales and the lactobacillales, with some key examples being bacillus species, which are found in the GI tract, helping to digest complex carbohydrates. We find Staphylococcus epidermidis, a common skin commensal that plays a key role in competitive inhibition, preventing pathogens from colonizing the skin. We also have lactobacillus species, which are common in the GI tract, where they aid in digestion and help maintain a healthy balance of microorganisms. And finally, there are different Streptococcus species which are abundant in the oral cavity and act through competitive inhibition to ward off potentially harmful bacteria. Next, we have the bacteroidetes. These bacteria are gram-negative, mostly rod-shaped, though a few cocci can also be found. They are primarily obligate anaerobes, so they thrive in oxygen-free environments like the latter parts of the GI tract, though some are facultative anaerobes, meaning they can tolerate small amounts of oxygen found further up the GI tract. Now, the most notable order within this phylum is the bacteroidales, which includes several important genera such as Prevotella species, which are found mainly in the oral cavity, where they help digest complex carbohydrates from our diet. The Bacteroides species play the same vital role in breaking down complex carbohydrates, however they are found in abundance in the GI tract, and it is here they start to produce short-chain fatty acids, which are key molecules that support gut health and influence our body weight and metabolism. Next, we have the actinobacteria, which are a more diverse group when it comes to the human microbiota. Now, they typically contain gram-positive bacteria with high GC content genomes, meaning their DNA has a higher proportion of guanine and cytosine bases. In addition to this, they show varied morphologies ranging from rods to filamentous forms, with most actinobacteria being aerobic, making them well suited to environments like the skin, although some are facultative anaerobes, able to adapt to lower oxygen conditions. Now, there are 4 notable genera, each with many important examples. So first up, we have bifidobacterium species such as Bifidobacterium bifidum. These are mainly found in the gut, where they digest complex carbohydrates and produce antimicrobial compounds that help inhibit exogenous opportunistic pathogens. Next are the various Propionibacterium species, most of which have been reclassified as Cutibacterium, for example Cutibacterium acnes, which is found on the skin, where they provide competitive inhibition against opportunistic pathogens. However, when they overgrow, they can contribute to problems themselves, such as acne. Then we have Streptomyces species, known for their ability to produce antimicrobial agents, and these bacteria play a key role in suppressing exogenous pathogens and are even the source of many clinically important antibiotics. And finally, we have Corynebacterium species, another common commensal of the skin, contributing to competitive inhibition and helping maintain the balance of the normal microbiota. And so overall, the actinobacteria are a functionally rich group, contributing to defense, stability and balance across multiple body sites. And finally, we have the proteobacteria, which are a large and highly diverse phylum that split into several major groups, including the beta, gamma, and epsilon proteobacteria. Now, some proteobacteria species are found as part of the normal human microbiota, particularly in the gut and urogenital tract, where they can play roles in nutrient cycling and maintaining balance within those environments. However, many proteobacteria are better known as exogenous pathogens responsible for a wide range of opportunistic infectious diseases. And so first up, we have the beta proteobacteria, where we find several important orders, including the Burkholdereales and the Neissereales. Now, the Burkholdereales include genera such as Burkholderia and Bordetella. Some species are harmless environmental organisms, whilst others, such as Burkholderia cepacia or Bordetella pertussis, can act as opportunistic pathogens, particularly in individuals with underlying respiratory conditions. Then we have the Neissereales, which include the genus Neisseria, which contains both commensal species and pathogenic species. For example, Neisseria meningitidis and Neisseria gonorrhea are responsible for meningitis and gonorrhea respectively. Next, we have the gamma proteobacteria, another major subgroup that includes several orders with both commensal and pathogenic species. Now, one of the most well known is the Enterobacteriales, which includes genera such as Escherichia and Klebsiella. Some of these, like Escherichia coli, are important members of the gut microbiota, aiding in nutrient metabolism and vitamin production. However, certain strains can become pathogenic, causing infections in the gut or urinary tract. Then we have the Pseudomonadales, which include Pseudomonas species such as Pseudomonas aeruginosa. These bacteria are often opportunistic pathogens commonly associated with infections in immunocompromised individuals or in cases where barriers are broken, such as burns or wounds. And finally, we have the Vibrionales, which includes various Vibrio species found mainly in aquatic environments. While some are harmless, others, such as Vibrio cholerae, are well-known pathogens capable of causing severe diarrheal disease when ingested through contaminated water. And the last phyla that we will look at are the epsilon proteobacteria. This group is smaller than the others, but includes some highly significant species associated with the GI tract. The two main genera are Campylobacter and Helicobacter. Now, Campylobacter species such as Campylobacter jejuni are common causes of foodborne infections, typically acquired through contaminated poultry or water. And the Helicobacter species such as Helicobacter pylori, which is perhaps the best known, can colonize the stomach lining, surviving the acidic environment, and in some individuals cause gastritis and peptic ulcers to form. And with that, we've looked at the main phyla and some examples that are either endogenous and helpers or exogenous and want to cause opportunistic infections. Now, as I mentioned earlier, the microbiota varies depending on the body site. Each location on or within the body provides different environmental conditions such as oxygen levels, pH, temperature, and moisture content, all of which influence which microorganisms can thrive in these environments. So let's go back through some specific body site examples. And I guess we'll start with the oral microbiota. Even within the mouth, the microbial community can differ depending on the microenvironment. For example, the teeth provide a hard surface for biofilm formation, while the gums and other soft tissues support different microbial populations. In addition to this, the composition of saliva also plays an important role in shaping which species can thrive. Now, overall, the dominant groups of the oral microbiota are the firmicutes, followed closely by the bacteroidetes and proteobacteria. And if we look at some key examples and their roles, we have Veillonella parvula, which helps maintain pH balance in the oral cavity by removing lactate and converting it into propionate and acetate. We have Streptococcus mitis, an early colonizer of tooth surfaces that helps initiate biofilm formation for other bacterial species. We have Prevotella melaninogenica, which helps break down protein, which is good, but as a consequence of this, it can lead to bad breath when they start to dominate the oral microbiota. And finally, we have Neisseria subflava, which readily consumes oxygen, helping to create anoxic microenvironments that allow select anaerobic populations to grow. So even within a single site like the mouth, there's a complex and finely balanced ecosystem of microorganisms, each performing different but complementary roles. Now let's look at the gastrointestinal microbiota, which varies along the length of the digestive tract. Now, this variation is influenced by factors such as pH, oxygen levels, and nutrient availability, all of which change significantly as you progress from the stomach at the start of the gastrointestinal tract to the colon at the end. Now, overall, the dominant groups of the gastrointestinal microbiota are the bacteroidetes, followed closely by the firmicutes and then the proteobacteria. And if we look at some key examples and their roles, we can see that we have quite a few. So Bacteroides fragilis plays a key role in modulating the immune system and maintaining gut homeostasis. We also have Bacteroides, the tau micron, which helps digest complex carbohydrates and break down dietary fibers, supporting nutrient absorption throughout the gut. We have Clostridium leptum, which is a major producer of butyrate, which is a short-chain fatty acid that fuels colonocytes and supports intestinal health. We have Lactobacillus acidophilus, which helps lower gut pH, creating an environment that inhibits the growth of various opportunistic pathogens. Escherichia coli often gets a bad rep, but in its commensal form, it produces vitamins and supports a balanced microbiota. And then finally, we have Enterobacteria cloacae, which can influence the metabolic environment of the gut. However, like some of the others, it can become an opportunistic pathogen if conditions allow. And so altogether, the gut microbiota forms a dynamic ecosystem that supports digestion, metabolism, and immune function, while helping to keep opportunistic pathogens in check. Next, we have the urogenital tract, and the microbiota varies depending on the anatomical site. For example, the vagina compared to the urethra, and it's strongly influenced by pH and hormonal fluctuations. Now, in the female urogenital tract, the microbiota is dominated by firmicutes, whereas proteobacteria dominate in males. And if we were to look at some examples for the female urogenital tract, we will see Lactobacillus species, which helps maintain low vaginal pH, creating an environment that inhibits pathogen growth, as well as both Staphylococcus and Streptococcus species, which are common before puberty, where they compete with potential pathogens and contribute to the early microbial balance before becoming less of an influence post-puberty. And finally, we arrive at our largest organ, the skin, which is home to a wide range of microorganisms, which varies depending on the type of skin, whether it's sebaceous, dry, or moist. And each of these environments provides different conditions that select for particular microbial communities. Now, overall, the actinobacteria dominate, followed by the firmicutes and then the proteobacteria. And if we were to look at some examples, Propionibacterium acnes, also known as Cutibacterium acnes, is commonly found on oily skin, producing lipases that break down sebum into fatty acids, which serve as nutrients for other microorganisms, which is good. However, when it overgrows, it can trigger inflammation and contribute to acne, which is bad. We then have Staphylococcus epidermidis, which is found across most areas of the skin, where it acts as a competitive inhibitor, producing antimicrobial compounds that help prevent pathogen colonization. And finally, we can have some Burkholdeриaceae species, typically found in moist environments like the underarms or groin. Now, these are usually transient colonizers, not permanent residents, but still part of the broader skin microbial community. And so with that, we come to the end of our whistle-stop tour of the human microbiota. We've only scratched the surface, focusing on the key phyla that dominate certain areas of the body. But the truth of the matter is that the actual composition of the human microbiota is extremely diverse. Each one of us carries a microbial world uniquely our own, constantly shifting, adapting, and evolving in response to our lives and the environment around us. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.